Sorry, guys. I forgot to sign in. It didn't ask me. I forgot to sign in to allow unlimited time. Okay. So now I can record a longer video, but we're almost done. Okay. So we stopped at, we're pumping hydrogen, right? So we're pump. We get more and more hydrogen on the outside. So naturally, these hydrogen ions will want to flow back into the cell, go into the negative charge, and go into the lower concentration of the hydrogen naturally, right? So that's what hydrogen is doing. But it's really nice because when hydrogen is doing this, it can also grab a friend. So in this case, we have a sucrose hydrogen ion cotransporter. So what it does is that even though hydrogen is going down its concentration gradient, it can allow sucrose to go against its concentration gradient. Okay? So it's kind of like you have a special, I don't know, you have a special registration with the country club, and then you can bring a friend kind of situation, right? So you're going there because naturally you want to go, right? You already paid, you've done your job, high to low concentration, that's great. But now you can bring a friend with you. And even though your friend didn't pay and is low to high concentration, you can bring it with you. So that's cotransporter, okay? So why does this matter? Why does this matter is because a lot of the times we want to have more and more sugar in certain types of cells, especially in plants, for example, right? So transporting of sugar in plants oftentimes need the incorporation of both the proton pumps and the cotransporter at the same time to allow more and more of the sucrose to get into the cell. Okay, so let's take a look at I guess this picture shows you the same thing, right? ATP allows proton pump to do active transport, low to high concentration, but then hydrogen is going high to low concentration. Sucrose is going low to high, but now it doesn't require any energy directly because there's already an energy, the electrochemical gradient is almost like an energy source. It's like, for example, water, right? Water going down, right? Water on its own, high to low, going from high to low, it's flowing that way at a bay or at a dam, is able to generate electricity, right? So as this gradient of water allows you to generate electricity. So similarly, the concentration and electrical chemical gradient of hydrogen ions is allowing sucrose to go from low to high concentration, even though there's no energy being used directly here. But there is energy used over here to allow this concentration gradient to be set up, okay? So another reason why this could be useful is that rehydration. So putting more solutes into your cell allows water to go into the cell as well. All right, this one right here shows you in our body, we have the epithelial cell in your intestines. So they're able to use cotransporter as well, right? So what you see over here, we have a sodium-potassium pump. Sodium-potassium pump is active transport. It does use ATP. It pumps a lot of the sodium out of the cell and potassium into the cell, as we saw earlier. So then what is the result? You will have a high concentration of sodium outside of the cell. You will have a positive charge outside of the cell. So naturally, if there is a protein channel being opened up, such as it is over here, then sodium can go from high to low concentration back into the cell, right? So you're pumping a lot of them out, and then they go back in this way. Low to high, and then high to low back this way. So while it's going high to low, this way it's allowing other molecules to be carried along with it. So in this case, it's going to carry glucose. But then for glucose, it's going to go to a low to high concentration. So you're building up more and more of the glucose in the cell. Okay? So low to high over here, and then high to low to high. This glucose can go from low to high because sodium is going from high to low. It's carrying a friend with it. And then after that, glucose can then passively transport through this channel and then get into your capillary and go into your bloodstream and go to the liver. Okay, so you can read these words on the left side. I'll give you a moment to read that. Right, so you can. What the point here is that you can see there are a lot of protein channels and carrier proteins and all that. They're all working together to allow the functions of the cells in your body. All right, moving on. Lastly, we're going to talk about endocytosis and exocytosis. So exocytosis requires energy because if you're moving this vesicle, it requires energy to direct it, right? So we can have proteins inside this vesicle. We're directing this vesicle to the cell membrane. It fuses with the cell membrane, allowing the molecules to go outside. So we've already seen this before. Examples, neurons releasing neurotransmitters. So you'll have a vesicle full of neurotransmitters, and it can fuse with the cell membrane or the neuron once it receives certain stimuli. Pancreatic cells secreting insulin or carbohydrates, whatever. Okay, and then the opposite of that, we have endocytosis, molecules coming into the cell. So these are, for example, big proteins. They're too big. They can't even, a protein can't go through a protein channel. It's too big, right? So in this case, these proteins can form a vesicle in the cell and then get carried into the cell. Okay, so that's endocytosis. Phagocytosis is an example of endocytosis. Here's a receptor-mediated endocytosis, just means you have these receptors accepting these signals and then bring these molecules into the cell. Endocytosis also allow bulk transport of specific material with low concentration in the environment. So for example, if you want to bring a lot of glucose into the cell, you could do that. Cholesterol for cell membrane synthesis. We can skip. So like, for example, you eat cholesterol, you are trying to get these cholesterol into your cell, you can do endocytosis. And then these endocytosis and exocytosis also remodel the cell membrane constantly, right? So you can think about when you're getting in exocytosis, the vesicle fused with the cell membrane, then now the cell membrane components are different now, or the opposite. Okay, that's all we have. Thank you, everybody.